Okay, everyone. So this is gonna be probably our last video of the semester, and I'm hoping um, that things will go smoothly. Lately, I've been having a lot of equipment issues, and so I'm hoping that it didn't happen like what it did in chapter 15, where certain segments of the video got corrupted. And so hopefully it goes well. But anyways, other than that, let's keep going. So we are now in our last two chapters. We've talked a lot this semester about the cell, and I know you guys are so sick of hearing, the central dogma, lipids, and all of that. But now we're really trying to give you guys that overarching perspective. And I do hope that you guys are able to take away, at least from this class, certain concepts that you can utilize in your future courses. Say that you're taking advanced molecular biology, then hopefully some of the concepts will carry over. Now, if you guys have taken physio, you'll notice that a lot of physio concept kind of melded into our biology here. And so, without further ado, let's keep going. And so now we're going to be talking about the segment of our cytoskeleton. Now, I always, for you guys, I always relate it back to what you've learned in Gen Bio and how Cell Bio is asking you guys to go a little bit more. And so in Gen Bio, we've taught you guys that for the cytoskeleton, there are three different types of filaments or three different types of structures that you guys are already know. And these are your microtubules, your microfilaments, like actin filaments, and then your intermediary filaments, and the location of which are also found throughout, the cell. And so, what you guys should already have in terms of a foundational understanding is which one is a bigger tube, which one is the smallest, the actin. Where are they located? What fibers are located near the nucleus? What fibers are located near the cell membrane? And those are the things that, again, these are background knowledge that you guys should already have from your past courses. Now, what's different, at least from Gen Bio to Cell Bio, is now we're going to go into a little bit more detail on the proteins involved on making these cytoskeletons. Are these cytoskeletons always present? And let me pull up my pointer here. Are these cytoskeletons always present in the cell? And if we're to move the skeletons around, can we move them around? And if so, do we move them all when they're fully formed, or do we have to move them piece by piece? And so these are the concepts that you guys should be starting to kind of be like, hey, these are the newer things. I don't know this stuff. And so we're just going to keep going from here. Okay. And so the first thing that we are, or the first filament that we are going to be looking at is the actin filament. Now, the actin filament is part of one of the most important filaments that you guys will know for this chapter. We will talk about the tubules, and the intermediary filaments are kind of like a subset of filaments that we aren't going to focus on too much. We'll talk about it, but it's mainly the actin and the microtubules that I want you guys to really maintain your focus on. Now, why is actin filament so important? Well, again, they're these filaments, these cytoskeleton, these cell skeletons. So cytoskeleton, again, is another way of saying cell skeleton, are involved in many biological process. The main one that we are going to be focusing on is muscle contraction. And your muscle fibers are all actin filaments, bind it up into a bundle and with a little bit of additional proteins surrounding those bundles. But they, in essence, are helical in shape. So we can see right here, just in the introductory slide of the actin filaments, we can see the size range, and we can also see that they are kind of turning in shape, very similar to that of a DNA. But what we also notice about these actin filaments is that they're made up of the exact same subunits. So the big thing here is that, hey, look, I can recognize this as a helical structure. They're all made of proteins and they're made of subunits. Great. Again, this is a repeat. In general, this is a repeat of like chapter 3, chapter 4 concept. Now, where do we go from here? Okay. Microtubules, same thing. These are all introductory slides. So microtubules, we can see there are same subunits based on the picture right here. But we can see that there are two different colorings versus in active filaments, there's only one. So chances are this is a singular subunit. And then when we're talking about microtubule, one, you can see a tube-like structure forming, but you also see two different colors. So these would represent two different subunits. And then if we look at the electron micrograph right here, here are a cross section of what a microtubule looks like. And you can see that in the center of this region right here, you can see a hollowed out portion. That hence why they're called microtubes. Okay. And so one thing that we are going to have to kind of visualize is how are these subunits pieced together and what orientation do we have? Now, if you guys pay attention to the bullet points, you'll note that I never really like to read off of the bullet points because I feel like you guys can do that on your own time. But these bullet points are a guide to the subsections that you would read in your textbook or the main overarching concept that you would need to know for the next several slides. So as we're looking through this, we'll notice that, hey, look, each of these subunits are going to be arranged from a positive to negative end, or in this case, a head to tail, to create a flexible filament. Now, we know, at least hopefully, we kind of have an understanding that if actin is our muscle fibers, our muscles are not stiff. They are very flexible. We can extend them, we can bend them, we can, we can squeeze them when during like a massage or something. But you'll know that it's not a hard, rigid like substance like your table, right? It's very flexible. So these microfilaments here, these actin molecules, are going to be extremely flexible, especially when we are piecing them together. Now, we notice on the slide right here, you'll see a plus N and a minus N. And here is just a 3D in-depth structure. We can see there's a lot of secondary structures found within this 3D tertiary structure. And we can see that ATP right here is going to play a very, very big role in actin. Now, for microtubules, we're working with GTP, not ATP. Okay, so actin, ATP, microtubules, GTP. Okay. And again, we have monomers versus a polymer. And so as we're looking at this, what you're going to see is that if we kind of take a look really closely, you're going to see that there are going to be a primary chain, which obviously is shown in the micrograph. But if you look slightly closer right here, you can start seeing that there's a V-like structure, something that kind of resembles this. And I don't know if my pen is going to show up very clearly, but here's the main chain right here. But the V-like structure kind of looks like this. And we can kind of actually tell the direction because of these branching structures that we see right here. The minus n will always contain the concave or the sharp point of this structure. And then the plus n will have the branching chain extending outwards. Now, this is what we call a barb-like structure. And so if we were to kind of analyze this graph and we're trying to figure out which side is positive, which side is negative, you'll have to look very closely to see where the barb-like structures are pointing towards and where all the points are pretty much confining in. And we can see that this side is going to be positive, and then this side is going to be the negative. Okay. All right. So here's the thing. Just like working with Lego pieces, you're going to have subunits. You're going to have the most basic monomer available. And again, this is going to be relaying back to chapter 2 when we were talking about the monomers of the different macromolecules, right? You have, your amino acid, a whole bunch of monomers together makes a polypeptide. For this case, the monomer is not necessarily the basic structure of the macromolecules. We're not talking about monomers of macromolecules right now. We're talking about monomers of the actin filament. And these actin subunits are those monomers. Now, unfortunately, to create a polyactin chain, okay, a polymer of actin, you're going to need to start building some sort of structure, some sort of base foundational structure for the actin filaments to kind of gather on. Now, when we are trying to build this oligomer, right, these multi-monomeric or multi-subunit structures, this is what we call the nucleation phase or the seeding phase. Now, we need to build a stable enough structure to know that, hey, this is stable for a long enough time where I can start building on top of it. Again, the nucleation phase is this positioning right here. So it will take a while. And because it's taking a while, this is also, if you take a look at the graph, this is a very similar graph to what you've learned before. Maybe you learned it in populational studies, or you learned it in, well, mainly populational studies. But we're not dealing with population right now, but this is a sigmoidal graph. Okay, so we will have a lag phase where it takes a while to get things started. Then we'll have a hyper growth phase or an exponential phase. And then we'll have a plateau phase or an equilibrium phase. So your oligomer, the foundational start, saying that actin has to grow by itself, once it has that foundational seed, then more actin filaments can build on top of it, growing into a longer chain. And this is where we hit the elongation phase or the growth phase, and the actin starts growing. And then after that, then it hits the steady state phase. Now, if you take a look, you're going to notice that on one end, you have a hypergrowth phase and multiple subunits can attach to it. On the other end, you're going to see that only one subunit can attach to it. Now, to keep things simple, we're always going to mention that the positive end is the faster growing side, and the negative end is the slower growing side, which we'll talk about in the next several slides. But once it hits a stabilizing form, just know that this process of attaching and releasing is occurring at an equilibrium rate. So you're going to add and remove at the same rate. And this is another way that we can call this a treadmill, right? Because a treadmill never runs out. It just keeps going on forever, never, never. But as we're adding subunits, we're also removing at the same rate, this each of those subunits. Okay, so that is graph A. Now, if we take a look at graph B right here, you're going to see that the starting position is when we already have a seed. Say that we just want to take a look right when the seed is formed. What does our graph look like? Or say that in your cell, you already have a baseline structure created elsewhere. Can the filament grow from that structure? And the answer is yes. As long as you have some sort of blueprint for these actin subunits to latch onto and begin growing, and it's stable enough, then we can just immediately go to the elongation phase, which is what this graph B is showing us. Okay. So here's the thing. Now, once you guys understand this, the idea of why is it or how does our cell know to kind of break the filament apart or add more subunits is through your nucleosides that you are working with. Now, like I said, you're going to be working with ATP and GTP. Now, depending on what filament you're looking at, if it's actin, we're going to be working with ATP. And if it's microtubules, we're going to be working with GTP. Now, for simplicity's sake, we are always going to say that the triphosphate is the more stabilized form. Again, when we're adding subunits in, they are in their triphosphate form, the ATP form embedded within the actin subunit. So if we take a look, again, here's your ATP embedded within your actin subunit. So if I was to use a different color here, I can highlight that the ATP might be embedded in this region where the blue dot is. Okay. Now this is your stable, more stabilized form. Now, as your actin subunits kind of treadmills through, okay, it then hydrolyzes and becomes a diphosphate, so ADP. Now, when it's in a diphosphate form, it's going to become more destabilized, so unstable. And if there's nothing preventing it from falling apart, say for instance, if I had like a little cap right here or a little structure that is a solid structure, if this is just free, then these diphosphates will begin breaking down. But if I do have a structure that is a very stable structure and it blocks the diphosphate from breaking apart, then this will stay rigid. It will stay set because we have a structure to kind of hold everything together. Okay. Now, again, dynamic instability and trembling are the main behaviors of the cytoskeletal polymers. Okay. Treadmilling, again, like a treadmill, it's going in a cycle. You're adding subunits on, and then you're taking subunits off equally at the same rate. Dynamic instability. Break the words down. Dynamic means things are moving. Instability means that there's a destabilization process. And it really depends on the rate of actin or microtubule subunits adding into the chain or removing the chain. And so the question is, when do we have a treadmilling state? Again, same idea. You're adding and removing at the exact same rate. Now, in this image right here, we're going to see that we're adding subunits and removing subunits at the same rate on the positive end. But again, as we keep doing this, we're adding newer and newer subunits on the other end, and this old subunit here is going to shift downwards through the chain. So here is the exact same subunits that we were adding and removing in the beginning. And then it's going to go down even more. So again, it's like a treadmill. It's going to treadmill, cycle around, treadmill, cycle around, treadmill, cycle around. And this is, in essence, why we're calling this a treadmilling process. Now, if the addition of subunits is greater than the releasing of subunits, we're going to have a growing effect. Okay. Disregard what's on the slide right now. What I really want you guys to understand is the dynamic instability right here. So if the rate of subunits added into the tube is greater than the subunits leaving, we have a growing behavior. This makes sense, right? If we're trying to relate it to what you guys already learned in Gen Bio through population, if you have a higher birth rate versus a declining death rate, you're going to have a growing population. This is the same concept here. And if we have a greater destabilizing process and a smaller growing process, we're going to have a shrinking behavior. Again, this is the dynamic instability of tubes and actins. Okay. So, microtubules, just know they depolymerize, so destabilizes, meaning disintegrate much faster, about 100 times faster from an end containing GDP, diphosphate, than one containing GTP. So again, the triphosphate is much, much better to hold into the structure. But over time, your body's going to need those phosphate groups. They're going to hydrolyze the phosphate within um, the cytoskeleton, and that will destabilize it. Okay. So as long as you have a cap, you're going to be able to maintain those subunits within that region. Okay. And again, this is just an example of treadmilling, shrinking, or declining. I wouldn't worry too much about what this slide is saying. Just know that depending on whether or not you have a cap or not, will determine at what concentration you will enter the treadmilling stage. Again, this equilibrium point represents the net adding and losing of subunits. Okay. So now what are we looking at? Well, in terms of the creation of actin, these are the proteins that I do want you guys to have a better understanding of. I want you to know that formin, the role of formin. I want you to know the role of thymosin. Okay. ARP23 complex, what is it involved in? Prolifin, and a little bit of the tryptomyosin and the capping protein. Now, why is that? Because one, we need to learn what the creation of actin, how actin is formed, and two, how does it play a role in muscle contraction? Okay. Oh yeah, and tropomodulin. Okay. Tropomodulin being your myosin cap. Okay. So really quickly, if we just take a look at the complexes here, right? Formin, as the name suggests, it helps form your actin. So you start with the little actin seed, the nucleation seed, and then formin will help grow it along. And you can have multiple formins working at the same time to grow a bundle. Great. ARP23 complex. The main purpose is to add nucleation sites to allow branching to occur. Again, your cytoskeleton is a branch-like network. Just like our skeleton, our ribs are branch-like. The cytoskeleton, the cell skeleton is going to be branch-like. And what mainly creates those branches is your ARP23 complex. Now, your prolifin, okay, or profilin, not prolifin, profilin is there to promote the forming of actin. Now, how do they help promote it? Well, they help bring the subunits closer to the formin so that we can start building. Okay, profilin promotes actin assembly. Okay. Now, tropomyosin and tropomodulin, I'm going to hold off on a little bit, but just know that tropomodulin is your negative end cap, and your capping protein or your cap Z is your positive end cap to prevent your actin filaments from degrading. If you want to take a look at what tropomyosin is, it's on the side. It wraps around the main actin filament. Okay? So if you need to, just write down an index card just to kind of have an idea of what each of these terminologies are. All right. So I'm not going to go over this slide because we just kind of went over it right here. So let's go a little bit further. And we can see that formin right here is a dimeric molecule. And we can see that the actin filament is growing faster on the plus end. Okay. Now, what we can notice is that formin has this hand-like structure, and when it's done placing a subunit in, it's going to zigzag, okay, upwards. And as it zigzags upwards, there is now an open region where another actin filament can plop in. And that's where this actin filament is going to plop in. And then it's going to zigzag upwards, where now you have another area that actin can bind to. Okay. So formin is there to kind of highlight, hey, actin subunits, this is where you can come in and start forming. And indeed, this is what happens. Now, you can see that formin, being a dimeric protein, is also attached to a cell membrane, to the plasma membrane right here. So it's a peripheral protein. Now, with any peripheral protein, keep in mind there is an active form, just like we've been talking about throughout the semester, and a deactivated form. Okay. Right now, formin is in its activated form where it's going to start building the cytoskeleton. We can see profilin attached directly to this subunit, bringing in closer where formin is found, and it's going to be placed into this open subunit region. Okay. And again, depending if you have a cap, right, it's going to maintain the growth rate or it's going to maintain the GTP or ATP subunits for a much longer time. Okay, so we can see capped population of filaments growth at the minus n only. Now, why is that? Again, we've capped the top. So that means no more subunits can be attached on the top end, only on the minus end. And we've already specified that the minus end grows much more slowly than the positive end. Now, if they were both uncapped, then you can grow on both ends at the same time. Okay. So here is your myosin. Now, myosin is another subset of proteins that will wrap around your actin bundle. So say that you have your actin bundle right here. Okay. You have your actin bundle. Okay. Your myosin is going to be found on the outside of it. And let me use a different color, say red. It's going to be found on the outside of it, and it's going to have a little uh, head-like structure here. Now, these head-like structures are going to be very important as we are going to talk about it during muscle contraction. But take note right here that you do have some light chain uh, myosin, and then you have your coiled over coiled alpha helix. Now, this coiled over coiled alpha helix is going to wrap around each of those subunits. Okay. And then you have your N terminus myosin head. Okay. Now, do note that this is a form of motor proteins. Now, we haven't really talked too heavily about the motor proteins in this class, but just know motor proteins are what helps things move within the cell. So, your kinesin is an example of a motor protein that walks along the actin or the intermediary filaments. Now, for our case, these motor proteins are directly attached to the cytoskeleton because they're there to move and contract the other filaments and squeeze them together during muscle contraction. So right here, you can see all the myosin heads wrapped around the main actin filament inside, which isn't shown, but there is going to be a region where it is completely barren. This is what we call a bare zone where no myosin head is found. Okay. So again, myosin head, myosin tail. Hopefully you guys are able to see that. Now, when we're dealing with the actin filament is going to be found on the outside. The myosin is going to directly attach to a region on those subunits. It's going to latch onto it fairly tightly, and depending on the activation or deactivation of those heads will determine if you're contracting or relaxing. Okay. So right here, I highly encourage you guys watching, different YouTube videos. There will be one that I will show you guys in lecture when we have the lecture, the day of the lecture. Sorry, I'm blanking out here. But here is a YouTube link that you guys can watch. Okay, it might not be the same one that I show, but for this section of the chapter, I recommend just watching a visual. It's so much more helpful, at least for my learning style. I'm a very visual person, so when I'm looking at other videos that kind of just meld everything together very nicely, it makes a lot of sense. I'd much rather watch a 4 or 5 minute video than to read the chapter and spend 20, 30 minutes on it. That's just my opinion. But again, if you do better reading the textbook, then by all means, you're more than welcome to read this stepwise behavior or stepwise pattern that we should see during the contraction. But to summarize it, what's going to happen is that the myosin head, if the actin filament has an opening where the myosin head is slightly attracted to, remember, back in chapter 4, chapter 5, we've talked about how proteins are interacting with each other. It's not because they're thinking about it, it's just that there is a region, a chemical region on the protein that intermolecular forces is able to take hold of and fit perfectly. And so this myosin head being attached into this actin filament, it's not going to be strongly attached. It has some sort of attraction, but it's not super strong. Not until ATP starts playing a role. Now, ATP, being a hyperphosphorylated group, is going to conformationally change your myosin structure, the myosin head structure. Okay. Now, with this myosin head structure, it's going to detach itself from actin. Again, the binding of actin with the myosin head is not that strong. Once ATP comes in, it changes the conformation enough where it's going to release your actin. And then as it's going through its process, it's going to get hydrolyzed somehow. Another enzyme comes in, dephosphorylates ATP to ADP. And when that happens, again, we're removing a phosphate group, which is very, very negative. When we remove that, the conformation of your myosin head changes again. And in this case, it changes so much where now that open spot on the actin is much more desirable. It's going to have a stronger affinity for it. It's going to latch onto it, and then it's going to pull it towards itself. This is what we call a power stroke. And it won't release until ATP comes back in, replaces your ADP. Okay. ATP comes in, replaces ADP, and then it's going to go through this whole cycle all over again. And this is the contraction of actin in relation to the myosin. Now, you can see that the myosin filament, this is a very thick filament, are wrapped together. Now, I believe in the other slide, I did say that there might be a chance that there might be some sort of actin found in here. That might not be the chance. Sorry, I misspoke. Okay, so it's just a whole bunch of myosin wrapped around together to form a thick filament. Okay, and we can see that there's going to be a lot of nuclei around because again we need a lot of control centers to make sure everything's moving smoothly, and then you can see these myofibril, which is your actin and myosin complexes. Okay, so here you have your myosin line, the thick filament right here of your myosin. Okay, and then you also have a zigzag Z disc, which is where the actin are binding to. Now you can see that it is a branch-like structure where you have different branching points right here. Let me pull my laser pointer out, it might be easier, where there's different branching points where actin can start forming. Now, as it contracts, you can see that the Z discs are going to get closer together while it's power stroking. Now, an example or another analogy, a metaphor for how the myosin head works is like playing tug of war, or more specifically, um, I don't want to say fishing, but imagine that you had a rope and you need to bring the rope to yourself. How do you do that? You're going to grab the rope with your hand and you're going to pull it towards yourself. But you can't pull it towards yourself indefinitely, right? You got to release the rope and then grab the rope from a further end and pull it towards you and keep doing it until all the rope comes to you. That's exactly how your myosin is going to work with your actin. Okay. Again, it's going to attach itself, grabbing onto the actin, pulling towards itself, release, grab it again, pull it towards, release. Okay. All right. So take a look right here. Now notice your actin isn't connected. It's not connected all the way from one end to the other end. Okay. But you have tropomodulin. Now, I did tell you guys what is the role of tropomodulin. The tropomodulin is just your negative prime cap. Okay. It's the cap to stop actin from forming longer. It's the minus end cap. Whenever you have a cap, you're stopping the activity of that region. Makes sense, right? It's like a pen cap. If I put a pen cap back onto my pen, I can no longer write with it because it's protected. Your plus end cap right here is preventing the positive end of your active filaments from growing. So now that we have two ends that are capped, this, even though the active filament becomes dephosphorylated and becomes ADP, it's going to stay in one position. It's not going to disintegrate too easily. Why is that? Again, we have two stable structures holding the whole filament together. Another way to think about that, imagine you had a long, big, clear tube. One end of the tube is covered. You put a whole bunch of marbles in that tube and cover the other end with a cap. Now you have marbles running throughout the tube. But if you remove the cap and tilt the tube slightly, all the marbles is going to fall out, right? Because there's nothing holding it together. So that's another way that you guys can think about it. Okay. Now, building on to what we talked about last chapter. I know it's a lot of overlap. Okay. Our calcium ion gate is now present again. Now, we learned the calcium ion gate through the GPCR cycle, through the pathway of GPCR. It is one of those secondary signals, right? Calcium is a secondary signal. Now, calcium's main role in this is for the contraction of your muscle. But what, and so here's a pop quiz, or yeah, pop quiz. What causes the calcium to get released. Now, again, recall back from chapter 15. You need a primary signal. What is that primary signal? Well, for the muscle cells, it is going to be your neurotransmitter. Your neurotransmitter, again, nerve cells, releases some sort of signal to tell calcium and your muscle, hey, we need contraction at this place. If it needs it right now, your calcium is going to get released from your sarcoplasmic reticulum, and then it's going to interact somehow with your actin and myosin head. So I'm hoping that that kind of gets you guys to kind of see the overlap here. Okay. Now, how does it work? Well, what happens is right here, calcium, once a signal is released, could be a hormone, but mainly it's a neurotransmitter. For this particular case, it's always a neurotransmitter. It's gonna relay its signal. It's gonna tell your sarcoplasmic reticulum, release the calcium because a voltage change has occurred. Calcium gets released, and calcium will interact with the protein calmodulin. Now, calmodulin, calcium complex, is going to interact heavily with your light chain, your myosin light chain. It's going to wrap around it and cause some sort of phosphorylation in the myosin. Okay? Now, in this regard, you're going to have a complex found directly onto your actin tropomyosin complex. Now, your tropomyosin, again, is wrapped around the actin, holding everything together. It's like that clear plastic tube. But your troponin complex, okay, is found on it as well. Now, you're going to have your I region, your C region, and your T region. I is your inhibitory, C is your calcium, and T is your tropo. I forgot the term to be honest. Tropo. I think it's troponin. Tropo something. I forgot. But what does this do? Well, if we don't want the myosin head to interact with the actin at all, say that we want the muscle to be at rest and not to be contracted, the inhibitory position is going to be present, right? If it inhibits it, myosin can no longer bind to this. Great. Your muscles are in a relaxed state. However, if we want myosin, the head of the myosin, to bind to this actin filament, we have to somehow remove this inhibitor right here. Now, how do we do that? Notice that we have calcium, C, right? This is where calcium is somehow going to interact, whether it's through calcium or calmodulin. That's, you guys can read it up, but it's going to interact and it's going to bind in this region. Now, because it's binding here, it's going to have a conformational change on this protein complex. And because of this conformational change, what's going to happen is the inhibitory position right here is going to get released, opening up the binding site for the myosin head to directly bind to. And if that's the case, all of those complexes are going to have an opening, which allows all the myosin head in the area, or at least the one that's in close proximity to this binding area, to bind to it to contract your muscles. Okay. Again, there's going to be another YouTube video. Go ahead and click on that YouTube video, watch it, or look for a better one, as a lot of them are going to be depending on your learning style. Okay. And so, with that regard, that's how myosin, tropomyosin, troponin, calcium plays a role inside the muscle cell. But they can also play a role outside the muscle cell in other various forms. Now, for this slide right here, again, this is just a way to kind of show you whether it's in its inactivated state or its activated state. Remember, the calmodulin is going to wrap around the myosin head, the light chain of the myosin head right here. And if it's phosphorylated, it's going to have binding sites that can directly bind to the actin filament. Okay. All right. So that's actin in a nutshell. Now we're going to go into the microtubules. Now, microtubules, very similar in process, except we have a tube-like structure and we have two subunits. We have your beta subunit and your alpha subunit. Your alpha subunit is the more negative subunit, and the beta subunit is going to be the positive subunit. And notice it's arranged from positive to negative, positive, negative, downwards. So positive, negative, positive, negative, positive, negative, like a AA battery. Okay. Again, same idea, dynamic instability. If we don't have the cap right here and the cap somehow goes missing, this is what we call a catastrophe because everything starts falling down like a house of cards. But if we're able to recover that phosphate group, that triphosphate group, right, and stop it from disintegrating, then it'll start growing again, which we call a rescue. So this slide is essentially telling you guys how the terms catastrophe and rescue is applied. Okay. Again, tubulin dimers are the subunits. You have your alpha and your beta. On your beta, that's where your guanine exchange is going to happen because again, we're working with GTP. Okay. So if you have GTP, the protofilament, which is the beginning prototype of the filament, right? Prototype means the type before the mass production. Um, is going to be in an orientation that's fairly straight. But once you get rid of that extra phosphate group, because you don't have that extra negative repulsion, it's going to start curving downwards. Now, you guys already know from your own experience, as a stick starts curving downwards, it's easier to snap off into different pieces. So, what's going to happen? If it curves downward too far, it's going to start snapping off, creating a disintegration effect. And for it to kind of latch back on, you're going to need to exchange this GDP to become GTP so that you can bind it back onto uh, the microtubule structure or the protofilament. Okay. Now, we know nucleation sites are a thing, or nucleation is a thing, but we can speed it up by having nucleation sites. Now, how do we do that? Well, certain proteins will have regions where you have the base foundation already made. Now, this base foundation is where our accessory proteins are going to be found and your gamma tubulin subunits. Now, notice there's beta tubulin, there's alpha tubulin, but there's also a gamma tubulin, which is that foundational layer that can be created ahead of time before we start building the microtubules. Now, this is more prevalent in areas that needs tubules to be added and disintegrated, right? Especially during cell division. Remember, your spindle fibers, okay, recall back from Gen Bio, spindle fibers are made out of microtubules. They're the things that move the chromosomes around. So right here, your centrioles will have these gamma tubulin rings around the peripheral, allowing microtubules to grow from it, allowing you guys to move the chromosomes throughout the cell during the metaphase, anaphase, telophase. Okay. And so this is essentially what these slides are showing. Yes, you can have other structures attached to the microtubules, just like actin with the tropomyosin. You can have MAP2, you can have tau attached directly to your microtubules to aid in the movement, okay, with motor proteins. Okay. Now, you will have certain signaling proteins that will tell us, hey, we gotta build more or we got to destabilize it. But if we take a look at the shape right here, you have kinesin 13, which is a catastrophic factor, protein factor. You can see that it's going to start curving your protofilaments. Now remember, the curves at the destabilizing effect, right? It prevents the stabilizing straight formation. Okay, if it's curved, we're able to snap it off in essence. So this aids in the depolymerization effect. Your XMAP215 right here, just another name for a protein. No idea why they call it 215, but you can see it's kind of like holding it in place. If anything, it's kind of like a guide to allow the subunits to latch on in a straight position as it moves down the growing chain. Okay. And here is an area where we might see it happening. Now, with intermediary filaments, again, it adds stability to animal cells. Here are some examples. No need to go into it too heavily. Again, we're not going to focus too heavy on it, but just know that the monomeric form is also a helical, and the dimeric form is coiled over coiled. Okay. And if we want to create a stronger filament, just like with any type of fiber, we're going to coil more coils around it to create strength. Okay. Again, these are just examples of how we can build the intermediary filaments into a much stronger fiber. Okay. And this is an example if there's a defect or a mutation in the keratin, which is the intermediary filament. In the keratin building, you can see the deformation of the cell. So originally we should have a nice uniform structure, but with that mutation or deformation, it's gonna go a little bit more haywire. Okay. And these last several bits right here, mechanistic wise, you have inactive wave NPF, you have your Rho protein, and you have GTP. Now, guess what Rho is? Rho is a protein that holds your guanine nucleoside. What's that role going to do? Well, again, anything that holds GTP or ATP is there to activate or deactivate something, right? So when we activate Rho, Rho is going to go in, activate our inactive wave MPF. The inactive wave MPF, due to its conformational change, because of this GTP, is going to open up and allow prolifin or profilin, sorry, to bring subunits in. Okay. Now, because that happens, okay, NPF, okay, formin being your NPF, okay, is now going to get activated. The inhibitory domain, which is locks up your formin, okay, unlocks itself, opening up your formin hands right here, the little hand shapes that allows actin to start building. Okay. And again, this is just an example of the different pathways that Rho GTP is going to affect, uh, depending on what type of, uh, not structure, but what type of function we want it to do. Okay. And that's it for chapter 16. Very simple, very easy. With chapter 17, I will be, let's see, how long have we been talking? We've been talking about 30-ish minutes. Can we finish this? You know what? For chapter 17, I will put it in a separate video. So, with that being said, I'll do one more video. I said this was going to be a combined video, but let's do one more video. Chapter 17 shouldn't be too long, maybe another 20-30 minutes in chapter 17. Let's just end it here, keep things organized, and then be on the lookout for the next chapter. All right. Take care, everybody.